Thanks, Ray. Good morning, good afternoon, everybody. Thank you very much for joining us today for a webinar on the new ThunderBolts Myeloid NGS Panel, a translational laboratory experience. We have two speakers today, Dr. Eric Padron from Moffitt Cancer Center, as well as Jeff Olson from RainDance Technologies. First is a video housekeeping. A video recording of this webinar will be available on our website next week. Please check the link at the bottom here for access to this video recording of this event. One disclaimer before we go forward. RainDance products are for research use only. They are not for use in diagnostic procedures. The agenda this morning will be we'll do a quick RainDance product overview. I will do a quick 5-minute overview presentation on that, and then I will turn it over to Jeff Olson, who will talk to you about the ThunderBolts Myeloid Panel development, and then turn it over to our featured speaker today, Dr. Eric Padron from Moffitt Cancer Center, who will be giving his talk on a translational laboratory panel experience using the Myeloid panel. And then we will get into the Q&A session. A quick overview of ThunderBolt of RainDance products. So we have three main product sections. One is the Thunderstorm system, which is our industrial scale, high throughput, highly automated NGS target enrichment, highly accurate system. We also have the Raindrop system. It's an ultra-sensitive digital PCR platform for both cell-free and tumor profiling. And finally, we have our ThunderBolts NGS system, which is the bulk of the presentation today. It's a somatic tumor profiling NGS enrichment system that we'll be talking about today. Specifically on the ThunderBolts, it is essentially a platform that democratizes NGS enrichment for all. For the very first time, we offer one of the most sensitive nucleic acid analysis platform that is now enabled for both enrichment on the NGS enrichment chemistry, as well as building a dropletized digital PCR system. For ThunderBolts, we are able to actually assay both solid tissue as well as liquid biopsies as input into enrichment using the ThunderBolts. The ThunderBolts NGS chemistry allows customers to profile as well as monitor variants within the tumor samples. And specifically, we have launched a solid tumor cancer panel last year. And earlier this year, we released an open source option. It is an option of consumables to allow customers the flexibility to build their own ThunderBolts assay for NGS analysis of their cancer samples. Today, we'll be talking about the new Myeloid panel. It's a heme malignancy panel that both Jeff and Eric will be giving you a good overview of today. Prior to building the ThunderBolts Myeloid panel, we reached out to the community of heme malignancy experts, and we formed a hematology oncology expert panel. And you can see quite a laundry list of exports across both the United States as well as Europe. We looked at prominent cancer experts in comprehensive cancer centers as well as cancer academic researchers from MSKCC through Moffitt, MLL, as well as academic research labs at UCSF and Johns Hopkins. One of the HEMONC panel members is Dr. Eric Padron, who will be given his lab's preliminary evaluation of this myeloid panel that the expert panel helped us actually put together. At this time, I will turn it over to Jeff Olson, who will walk you through the ThunderBolts Myeloid Panel overview. First, a quick bio for Jeff. Jeff is the Director of Genomic Assay Development at RainDance, has worked in the biotech industry for the past 30 years. His experience includes early-stage startup to large pharmaceutical companies. Jeff has been with RainDance since 2009. He's worked in various projects on the development of several microfluidics-based assays. In his current role as the director and leader of the genomic assay team, he is responsible for building the entire ThunderBolts NGS portfolio of assays, both on the cancer panel as well as the new myeloid panel, which he's going to be talking about today. Jeff graduated with a degree in biology from MIT. Jeff?
All right, thank you, Omo. I'm going to give a quick overview of the ThunderBolts application and then present some data on performance metrics for the Myeloid panel for internal testing and customer data, looking at sequencing coverage depth, uniformity, and the detection of low-frequency variants. After that, I'll turn it over to Dr. Padron to describe the work that's being carried out in his lab. As Omo already mentioned, the ThunderBolts Myeloid panel consists of 49 target genes, shown here in this slide, which were selected in collaboration with a consortium of key leaders in the field of myeloid cancer. 15 of the 49 genes have full coverage of all exons, and 34 genes have coverage of select exons. There are 548 amplicons in the panel, and it covers 126 kb of target sequence. After generation and optimization of the myeloid panel, we tested it internally with 64 runs of Coriell DNA samples to evaluate its performance. We wanted not only to test for sequencing coverage and uniformity of the amplicons, but also for the ability to detect low-frequency variants. To accomplish this, we first sequenced 3 Coriell samples, shown here, to identify variant differences between the samples. We then generated a blend of Coriell samples at different percentages to simulate the presence of 35 variants at allele frequencies anywhere from 2% to 12%, using the Coriell sample 18858 as the background sample and Coriell samples 11831 and 19240 as the spike-ins to generate those low-frequency variants. Also, to stress the system, we used our lower limit of 10 nanograms of input DNA, which was also shared to 500 base pairs to simulate fragmented DNA like you might see in FFPE samples. The 64 samples were processed 16 samples at a time on the MiSeq using both the V2 and the V3 sequencing kits. And then the sequencing data was analyzed to evaluate coverage, uniformity, and for variant detection. This slide outlines the basic principles of the ThunderBolts application. First, you start out with a sample which contains the target molecules of interest, non-target background DNA, PCR primers specific for the target regions, and the PCR reagents. The samples are then loaded into a microfluidic chip and then placed on the Raindrop Thunderstorm source instrument. The source instrument then generates 16 million 5 picoliter volume droplets. And what this does is it partitions those target DNA molecules so that each droplet contains only one target molecule. For instance, in this example on the slide, if you start out with 10 nanograms of DNA, which is approximately 3,000 genomic equivalents, and you multiply that by the number of amplicons in the panel, you end up with about 1.6 million positive reactions. If you divide this by the 16 million droplets generated by the source instrument, you end up with one target per 10 droplets. RainDance's ability to generate millions of droplets allows us to do truly digital PCR. Now, there are advantages of partitioning each target molecule in its own droplet. Because each droplet has only one target molecule, there is no competition between amplicons. Competition between amplicons in the same reaction can result in sequencing read bias, in which a well-performing amplicon outcompetes a poor-performing amplicon. Also, because the reactions are in a precise, confined volume, each of the reactions goes to a very consistent endpoint. Both these factors result in the uniform amplicon yield between targets, which results in an even distribution of signature reads for amplicon, which you'll see in the data I'll present below. All this can be accomplished with a simple time-efficient workflow. And as you see here in this example, if you're processing 8 samples, the hands-on time is less than 3 hours, and the turnaround time from sample to sequencing data is about 2.5 days. This slide shows the results of 16 samples looking at the sequencing coverage depth for all amplicons and the percentage of amplicons with coverage at 0.2x of the mean or greater. As you can see in this top panel up here, the coverage is excellent, with 99% of the amplicons having 500x coverage at 5,000 mean amplicon reads. The uniformity shown in this panel here is also very good. All samples have greater than 95% of the amplicons with reads at 0.2x of the mean or greater. The coverage provided by the ThunderBolts Myeloid Panel ensures that you'll receive reads for all your target regions of interest, and the uniformity will allow you to multiplex more samples per MiSeq run. The data can also be shown graphically. This slide shows the coverage plot generated by the Myeloid panel at different coverage depths, average over 16 samples, going from 1x coverage all the way to 500x coverage. This slide shows the percentage of samples with coverage at 100x. The Myeloid panel consistently achieves 100x coverage for 100% of the amplicons and 95% of the amplicons at a coverage of 500x when the data is normalized to 2,500 mean reads. These lines show the coverage data obtained at 3 different beta sites where we sent our panel for them to look at. What this demonstrates is not only can we obtain consistently good results in RainDance, but our customers can also achieve the same level of performance. This light blue line is the coverage data for the same set of samples run using the Truesight Myeloid panel processed at one of our beta sites. This data demonstrates that the Myeloid panel provides superior coverage when compared to a Truesight Myeloid panel. The data on this slide was provided to us by Dr. Torsten Haverlock at the Munich Leukemia Laboratory. The 2 panels in this slide show the sequencing reads obtained for genes CEP alpha and RUDX1. Excuse me. Each bar represents the sequencing reads for single amplicons for each set of amplicons for each gene. The red and gray portions of each bar represent the forward and reverse reads, and the black line represents the average reads per amplicon for the set of amplicons for each gene. These are genes that are very important in myeloid cancer, but they're also notoriously hard to amplify due to regions of high GC. Even though these are tough regions, they're able to successfully use the ThunderBolts Myeloid panel to amplify them to a sequence depth coverage of 800x or greater. As I mentioned earlier, we made a blend of Coriell samples to simulate the presence of low-frequency variants. We used sample 18858 as the background sample, or our normal sample, into which we spiked the other 2 cell lines. We sequenced the 18858 sample by itself and also the blend of 3 Coriell samples and looked for the presence of the expected 35 low-frequency variant sites. A cutoff of 1% was used to make a positive call. The results for the 32 samples, the 16 background 18858 and 16 blends, are shown in the table on this slide. If a positive call was made in the 18858 sample, shown in this column, it was labeled as a false positive because it being the background or normal sample, it shouldn't have any variance. If a call was made in the blend, shown in this column, it was labeled as a true positive because these samples contain the spike-in DNA to generate simulated variances for the low allele frequencies. From that, the positive predictive value, shown in this column here, was calculated by taking the true positives over the sum of the true positives and the false positives. Then the numbers in these columns were the number of missed calls for the false negative or false negatives for the predicted 35 SNV in these samples. So when analyzing the samples using the background or normal sample versus the low variant frequency blend, we get a very high positive predictive value of 98.4% and a very low false negative rate of 2.3%, demonstrating that we can detect low frequency variance down to 2% using this method. Not only did we want to demonstrate the ability to detect low frequency variance, but we also wanted to verify that the variants were in the expected allele frequencies. This slide shows the plot of the expected minor allele frequencies shown on the x-axis versus the observed minor allele frequencies on the y-axis. As you can see, there is a very good agreement with the expected minor allele frequency and the observed minor allele frequency with a correlation coefficient of 0.9446. This table contains comparison between the ThunderBolts Myeloid panel and the Illumina Truesight Myeloid panel. The ThunderBolts Myeloid panel contains 49 genes covering 126 kb of target region, while the Truesight Myeloid panel contains 54 genes consisting of 141 kb of target region. ThunderBolts has a superior coverage with 100% of the applicants with 100x coverage or greater at 2,500 mean reads, and 99% coverage at 500x with 5,000x mean reads, while Truesight only has 95% of the applicants at 500x or greater with 5,000 mean reads. The Thunderbolt panel achieves good uniformity with 97% of the applicants with reads at 0.2x of the mean or greater, with typical results closer to 97%. As shown in the data presented earlier, we can detect minor allele frequencies of 2%, where Illumina claims only 5%. Also, the ThunderBolts panel requires only 10 nanograms of input DNA compared to the 50 nanograms needed for the Truesight panel, which allows research to stretch the pressure of samples further. Because of its high uniformity, the ThunderBolts panel can process greater number of samples per MiSeq run than was recommended by Illumina. For instance, for the V2 kit, we can process 8 samples. For the V3 kit, we can process 16 samples, where Illumina recommends 4 samples for the V2 and 8 samples for the V3. RainDance also sells the kits in 2 different configurations, 24 and 48 samples, as opposed to just the 96 samples for Truesight, allowing researchers flexibility in their experimental design. This slide has a summary of the specifications for the ThunderStone Myeloid panel. Because I covered most of this in the last slide, I won't go over it again. However, I want to point out something that was not covered or hasn't been covered yet, and that's the NextGene Software. The NextGene Software enables biologists with a limited amount of bioinformatics experience to analyze next-generation sequencing data. RainDance worked with Genetics to add specific modules to their NextGene platform that are designed for the analysis of data generated by the ThunderBolts Cancer Panel and the ThunderBolts Myeloid panel. The software can take the data all the way from FASTQ files, which come directly off the sequencer, all the way to VCF files containing all the variants detected in each sample. The NextGene software is an excellent tool that can be used by both experienced and inexperienced biologists to analyze data generated by ThunderBolts Cancer and Myeloid panels. I'm now going to turn it back over to Omo, who will introduce Dr. Padron.
Thanks, Jeff. We are now going to go to our featured speaker this morning, Dr. Eric Padron, whose talk will be on the translational laboratory experience using the ThunderBolts Myeloid panel. Quick bio on Eric. Eric Padron is currently an assistant member in the immunology program and malignant hematology department at the H. Lee Moffitt Cancer Center. After completing his undergrad medical school and residence at the University of Florida, he joined Moffitt Cancer Center for his hematology and oncology fellowship. Today, his current research interests are focused on understanding the genetic pathobiology of PMML using primary patient samples to understand the clinical implementation of genomics and to generate novel therapies in CMML and other myeloid malignancies. Eric?
Thank you very much, Omo. It's a pleasure to speak this morning, and I'd like to extend a warm welcome as well as a good morning and good afternoon to the audience. I thought before I'd start that I'd give a sort of framework of what our lab is and what our lab is not, so that you can take our experience in context. And so what we are not is a large genome-wide association study laboratory. We're not a clinical genomics laboratory, and I'd like to emphasize that none of the data that I will be showing today is meant to imply that that this test is a CLIA-certified test or should be implemented in patient care. And we're not a genomics core where we're processing samples for the entire institution. We have a great bioinformatics collaboration, but we, at the heart, are not bioinformaticians. What we are is an NGS, or I like to think we are, an NGS-savvy lab. We recognize the importance of using these types of panels in the disease we study, which I'll get into in a second. And many, if not all of our assays that we do in our laboratory converge on target enrichment sequencing. And so with that framework, I'd like to give you a brief kind of overview of our pilot experience using the Thunderbolt panel. So why are we interested in these targeted NGS panels? And the reason I think is summarized nicely in this slide from Lawrence et al. And what I'd like you to focus on is the relative genetic homogeny of acute myeloid leukemia, and by extension, the rest of the myeloid malignancies, and that compared to carcinogenic tumors like lung cancers and melanoma, AML and other myeloid neoplasms have relatively few driver mutations that in part can be captured by a myeloid panel like the one that RainDance offers. So a little bit about our model for context. So the disease we study is chronic myelomonocytic leukemia, or CMML. This disease is interesting to us because it really stands at the bridge between two important diseases of the bone marrow. One is a bone marrow failure disease called MDS, and another one is a myeloproliferation disease called MPNs, and there's a series of them. So CMML is the most frequent overlap syndrome disease, and we think in studying this disease, not only can we improve the clinical care of patients with it, but we also think we can learn a lot about the biology of the diseases for which it encompasses. So what is CMML? It's a disease that by definition requires a monocytic predominance in the peripheral blood. It's a disease in where symptoms are a huge component of the morbidity that can include splenomegaly or constitutional symptoms like fevers, weight loss, and night sweats. But important for this talk, the genetic fingerprint, as I like to call it, of CMML is actually very well defined for a rare malignancy. And so we know what the driver mutations are. In fact, there was a paper out of Dr. Hofferlich's laboratory that showed if you sequence only 9 genes, that you would be able to find 1 mutation in this disease in 93% of cases. And I think most importantly, what we know about this disease is that it's a very aggressive disease. There are no therapies. And so we think this is a nice model to study the genetics of myeloid neoplasms, and we think it's an important one because of how aggressive it is. So how do we use these NGS myeloid panels in our laboratory? Well, we in general do 2 things. One thing we do is take viable cells, primary patient samples or cell lines, depending on the assays we're doing. We manipulate them in vitro, and then we use, for instance, the ThunderBolts RainDance panel as our readout to understand what occurs after these in vitro stimulations to the composition of these important 49 genes, both the loss of these genes or the enrichment of other genes that we perhaps wouldn't have seen if in vitro manipulation wasn't done. One very challenging aspect of this approach, however, is that if you do this in a 96-well format, for example, you're dealing with relatively low input amount in terms of genomic template. And so we believe that's one challenge, and we'll talk about how the RainDance system may help improve that. And then the second thing we do is that in our disease, bone marrow biopsies are actually common. And because it is rare, we really want to leverage the tissue that has been collected prior. And in our institution, we're fortunate in that we have a total cancer care IRB-approved protocol whereby if patients consent, we can actually leverage this tissue to understand more about their disease. And so a lot of what we do is take this FFPE, perform the myeloid sequencing panel, and then associate it with clinical outcomes, survival, leukemia-free survival, phenotype differences. But the problem here is that there are, as many of you know, FFPE results in poor quality DNA, and actually what I think is more challenging, especially in an amplicon-based panel, is these deamination reactions that result in transitions from, for instance, a cytosine to thymidine. And I'll show you in a little while how these can actually be very important in terms of variant calling. Okay, so one of the highlights that I want to bring up with respect to FFP is the fact that when you compare, for instance, cell line fresh samples to those of FFPE, whether you restrict it to allelic frequencies of 1 to 10 or 10 to 25, you recognize that these C to T changes are much more common, and these, as we all know, are a result of these deamination reactions. And one thing that I'd like to highlight that's important clinically is that even though most of these C to T transitions can result in artifactual mutations that can be removed after bioinformatic analysis, some of these can actually result in what could appear to be actual driver mutations. So this is, for instance, a sample published in BMC Medical Genomics where a primary patient sample was found to have an NRAS G12D mutation in an FFPE sample. But after treating with UDG, which cleaves deamination reactions, the NRAS G12D mutation went away, suggesting that it was an artifactual mutation that could have been called as a driver event. And so this is a huge challenge with respect to FFPE tissue. And when you do this in an amplicon-based targeted panel, especially when the amplicons are pooled, it's very difficult to discern what is amplification of artifact and what is true mutation. So basically, to summarize why we use these and how we use this, our lab is in a unique position where we collaborate with our genomics and informatics core, but still have a hands-on experience. So on the front end, our laboratory generates the libraries, in this pilot case using the RainDance system. We deposit them in the core where they perform the sequencing. But then on the back end, because we don't have full-time bioinformaticians in our laboratory, we really do rely on commercial suites like Partex Genomics, QIAGEN GeneRead, and CLC Bio in collaboration with bioinformatician consultation. So that end-to-end NGS experience is very important to us, being a small laboratory. So what has been our experience with other panels? So one panel that we had used extensively was a QIAGEN custom GeneRead panel. And the reason we used this is because it was a low-cost initial investment for a 26-gene custom panel that essentially covered all of the genes tested. It required 100 nanograms of input, which we thought at the time was okay. And it had end-to-end support. And I'd like to emphasize how important this is for our lab. But the end support is not customizable. It's a web-based tool. You plug in your FASTQ files, and you get out a a variant list based on the specs and the filter strategies that are already embedded. So how long did this library take to make? So we've had extensive experience with this, and just to quickly go through it, there's a DNA quantification step, there's an amplicon enrichment step, adapters and indices, there's a purification step, then a bioanalyzer step, and then a library quantification step, which altogether resulted in over 6 hours of total hands-on time. The results of this panel were, I would say, adequate. There was expected dropout of difficult to capture amplicon, or difficult to capture genes like CDBC alpha and RUNX1. This was the median coverage for reference, so that you can see how this panel in our hands compared to the Thunderbolt panel. And so a little bit about why we thought the RainDance system may be beneficial to us. And I'd like to refer you back to the two challenges that we talked about, the low input and the FFPE deamination reactions. Well, in discussing with RainDance, because of the methods that were discussed earlier with respect to how the libraries are created, the fact that only 10 nanograms of sample is required was huge for us, because it allowed for those 96 well experiments to be done with confidence. The cost was actually lower, and importantly, because the reactions occur in kind of a pseudo-single plex fashion, our hypothesis is that perhaps the amplification of deamination reactions would be minimized, and therefore we could kind of remove those by filtering out variants that occurred at very low allelic frequency. We also like the fact that there's the potential for higher throughput with their Thunderstorm machine, and we're actually very excited about their collaboration with Soft Genetics and NextGene, and in fact, all the data you'll see here was analyzed with that NextGene tool. We're excited about this tool because, unlike the previous tool I mentioned, it actually is customizable. So it has the ease of use that the web tool had, but we can change things as we like in collaboration with Soft Genetics, so we thought that was very important. So I'm not going to go over this, but when we say single plex, what we mean is that the droplet generation allows for one genomic template to interact with the primer pools. And the hypothesis, at least in our lab, was that this methodology may allow equal amplification of genomic template and minimize amplification of deamination reactions. So what was our experience with the workflow? Well, the short description is it was much quicker. In our lab, our library prep technician routinely can do this now in 2 days, very comfortably. I like to point out that there's no library quantification at the end, and the total hands-on time was 2.25 hours, and this is reproducible on a 4 sample per droplet run basis. So one thing that we were very excited about is when we looked at the libraries by Bioanalyzer, we saw exactly what we expected to see based on the product description given by RainDance. So we see a peak here, which represents the large amount of amplified target region, and this non-specific peak here in both pools of libraries. And so that brings me to the point that in this panel, you actually have two PCR pools that are put together on the sequencer, and both of them performed excellent with 50 nanograms of input, so half of what we were used to putting in. What we were actually more excited about is when we saw the FFPE samples, whose libraries looked identical to that of the fresh samples, which was something we were not used to, again, with only 50 nanograms of input. And so we, at least on the level of the bioanalyzer, were unable to distinguish any differences between FFPE DNA and fresh DNA, and that was very exciting to us. So what about low input? Well, this is our pilot experience, and what we found is that we were able to push it in some instances as low as 15 nanograms, and this is on our first run without any optimization. We were able to have, we tried 10 and even 5 nanograms, and because it is 2 library pools, in some instances we were able to get amplifiable libraries, and others not. So we only went ahead and sequenced those in which both libraries had amplifiable both libraries had amplifiable DNA. And so in the first run, we were able to go as low as 15 nanograms, which was, again, very exciting for us, given the nature of our in vitro experiments. Another thing that was nice to see was, and again, all of this data is pulled out of either NextGene or the MiSeq software itself, that even without library quantification, we were able to see a relatively evenly distributed run among the samples, and this brings up how many we multiplexed. In this assay, we multiplexed a total of 20 samples together. So these are our statistics in terms of reads on target. These are our total reads. What I like to point out is that the forward and reverse reads seem to be superimposable, which is nice to see. The percent reads on target was 97. Our average coverage, multiplexing 20 deep, is 2,878, which was great, actually a little bit higher than what we were doing with the other panel. And the percent of our target, which was covered by more than 500x, was 95%. So these are all metrics that actually surpassed that of our previous panel. So again, we were excited to see that. This is the amplicon distribution. And so you can see what our average coverage was, but you can also see that many of these amplicons reach much higher. And I cut it off at 3,000x, but there were some that were much higher, even up to 11,000x. Now, there was some dropout in this beta test for CBP alpha. And ASXL1, interestingly, this was comforting for me to see, because again, all of this was done with the NextGene software, is that we got no coverage of this ASXL1 amplicon, and when we went back, we found that amplicon was actually missing in our pool. So this was very comforting for a physician and biologist to see, who is not a bioinformatician, that the NextGene software was actually working quite well. When we compared metrics between FFPE and FRESH, we did see a slight increase in the total reads, and as a result, a slight increase in the average coverage. But again, what I was very happy to see is that the percent of reads that were on target were almost identical, and the percent of regions of interest that were covered by more than 500x was very similar among FFPE and FRESH tissue. Lastly, the transition to transversion ratio, which as many of you know, is associated with those FFPE sequencing artifacts, was slightly higher in the FFPE compared to the fresh tissue, but based on what we have seen previously, this is only a slight increase compared to other panels that we've used. So we think that this pilot data may be suggestive that our initial hypothesis that this single-plex reaction is actually better for the FFPE samples may indeed be the case. So 2 conclusions. The first is that we were able to generate in our pilot experience good quality libraries from both fresh and FFPE from as low as 10 nanograms, although we inputted 50 nanograms, which is half of what we were used to and had no problem. What I really like and what our library technician really likes is that it's quick, and that they're sustainable library preps, and they can be done in 2 days very comfortably, manually, and that the consumables are low cost. One thing that we actually would like is that we think that the droplet generation is very important, and I know this is something that RainDance is working on, but increasing the number of samples that could be done on the droplet generator would be an ideal step. And I know this is something that they're working on, but it's something that we think is so important for the library prep that we'll be happy to see that. And lastly, we were, at least at the level of the Bioanalyzer, were unable to distinguish any differences between FFPE libraries and fresh frozen libraries. We saw a very low increase in the C to T deamination reaction. And we think that amplicon-based FFPE capture using this panel is feasible, and it actually may improve if we drop the allelic frequency to 10% in FFPE so that we can remove those artifacts. And of course, this is our pilot experiences, so further study is needed. We're actually performing an FFPE project where we're multiplexing 42 deep using, again, FFPE samples, and have several other projects in the pipeline which we're excited to move forward with. So I'll move back here, and I'd like to, that's the end of my presentation. I'd like to thank OMO and RainDance for allowing me to test their pilot, Myeloid panel, and, we'll take any questions at the end.
Thank you very much, Eric. Wonderful presentation. We have quite a number of questions, so I'm just going to run through these questions fairly quickly. I will call out the question, and then I will call out the responder to the question. Jeff, this is a question for you. How long does the actual droplet generation process take for a single 8-well chip?
It takes about 30 minutes to generate the droplets.
Thanks, Eric. Thanks, Jeff. There's another question here. This one is actually directed to me. When is the Myeloid panel commercially available? Is it available today? Yes. This panel was launched at AACR a couple of weeks ago, and it's currently shipping. So if you need to order one, contact your sales rep. The next question is for Jeff. What is RainDance recommending for sample indexing on a single flow cell of the MiSeq?
So we're recommending 8 samples if you have a version 2 kit for the MiSeq, and 16 samples for the version 3 kit.
Okay. Thanks, Jeff. Another question, this one is for Jeff. Is sharing of target DNA necessary to get single target inclusion in the droplet?
Yes, we do recommend sharing probably from 500 base pairs to 3 kb because it is breaking up the DNA. Some of the amplicons are close to each other in the genome. We do take care of part of that because we have 2 poles, set 1 and set 2, where we specifically separated amplicons that they were either overlapping or close to one another. So you can actually get good results if you even use unshared DNA. But probably for the best results, the sharing that 500 base pair to 3 kb level is recommended.
Another question for Jeff and maybe even Omo as well. Do you have any comparison data with the AmpliTeq AML assay from Life Technologies?
At this time, we do not. We have done testing in the past with other panels looking at AmpliTeq versus our panels where we perform favorably, but we don't have any for this particular panel.
So to paraphrase Jeff, on our cancer panel, we have a direct head-to-head comparison between the LifeSeq cancer panel, cancer hotspot panel, and the RainDance cancer panel, but we have not done the same for the Myeloid at this time. One more question. This one is for me. What is the Thunderbolt system, the Thunderbolt NGS system, is it the same as the raindrop source instrument for digital PCR? The answer is yes. So you have a single box that can not only enrich multi-gene targets for NGS screening, it can also dropletize for low-plex digital PCR assays, same box. All right, so one other question. This one is for Jeff. What is the minimum recommended input DNA to run the Thunderbolts Myeloid panel?
We've gone as low as 10 nanograms of amplifiable DNA, and that is, with various FFP samples and normal genomic DNA.
Thanks, Jeff. So we don't have any more questions. I'm waiting if there are any additional questions, and we'll wait to see any more questions. So I have a question for Eric. So, Eric, your preliminary study, you added 20 samples for a run. Is that your typical throughput you project to run, or do you intend to index less number of samples for flow cell?
I mean, I think our because we're performing, research grade targeted sequencing, we actually hope to get it up to 42 and even 48 on a typical MiSeq run. What we'd really love to do, especially in those 96 well experiments, is to be able to, in a couple of runs, take a full 96 well plate to NGS, and so I'm hoping, and I'm confident that we'll be able to get even higher.
All right. Well, in the absence of any further questions, I want to thank Jeff Olson, and I want to thank especially our featured speaker, Dr. Eric Padron from Moffitt Cancer Center, who has taken time out of his very, very busy schedule to join us and put together his lab's work on the ThunderBolts Myeloid panel. And I want to thank every one of you for taking time out of your very busy time to join us. Thank you very, very much. Again, a recording of this webinar will be available on our website beginning of next week. Come back and check us out anytime. Thank you so much, and have a wonderful day.